utility and powerful and
Good morning, everyone. So today is the third lecture of this introduction to PKPD. So I will resume what we were saying yesterday. So remember, we are in the context of brain tumors, and we are interested in glioblastoma, which is the most frequent and most aggressive brain tumor in cancer, in adults. So we are interested in temozolomide, which is the only drug, or the main drug that is used in the treatment of glioblastoma. So what we have learned today is that it was possible to design a mathematical model, I will go directly to this one, a mathematical model of the pharmacokinetics of the drug at the cellular level. So where we do have two compartments, so one for the extracellular space and one for the intracellular space. And remember that what we have seen is that basically temozolomide, TMZ, is activated in a two-step process. There are two steps. First step is going from temozolomide to MTIC, and then MTIC to AIC. And this meeting pattern, that is the most important species that will actually create the image and then cell death. So this is what we have seen is that this is a purely ODE model, just like what we have seen from the beginning of this lecture. And here the idea is to extend this study to account for space. So this is what I'm going to talk about today. We will address the point number 2 and number 3 today. So here I would like to present you a hybrid model that is a space model that will combine ODEs and PDEs, spatial PDEs. So the domain will be a 2D domain, which is grid. So we have 200 by 200 grid element grid. So at each node of the grid, you need to imagine that there may be a cell. So let me draw this for you. Okay, so this is your domain. At the node, you may have cells. For instance, here I can draw cells. Okay, cells. And basically, at each node here, each special node, we will put one system of ODE. So for each cell, we will solve one ODE. So then, for the rest of the grid, we will assume that the nodes where there is no cell, there will be a vessel to provide oxygen, because the cells need oxygen to live. So basically, this is a 2D representation, but you can imagine this, for instance, as the slice of a tumor. If you have a 3D tumor, you want to cut it like that, okay? And the idea is that basically you have the slice, and actually in the 3D you have blood vessels, and this would be the point that we actually represent, where we represent the blood vessel, and this is my grid. This is the cells. And inside the blood vessels, we have the oxygen. So the oxygen will be represented with the PDE, reaction diffusion PDE, which is the one that is described here. So it's actually quite classical reaction diffusion equation. The first term is diffusion. So oxygen is allowed to diffuse throughout the domain. With T0 the constant, the coefficient of diffusion. Then the second term is for the transport of the oxygen from the blood vessels and towards blood vessels. So basically, OV, this OV, is the concentration of the oxygen in the blood vessels. Maybe I will write this. And it's supposed to be constant because basically the body is able to provide oxygen, so it's supposed to be constant. So basically the second term is just the application of the Fick law that we have seen yesterday and the day before, where you have oxygen that will go towards the cells and that will follow basically the difference between the concentration. So O will be concentration at the spatial point, and OV will be the concentration in the blood vessels. And basically, this VIG is, maybe I should have said that CIG are the cells. So basically, here my coordinates would be, here I will be IG, okay? And CIG will just be equal to 1 if there is a cell, and equal to 0 if not. And VIG is just a complement. So whenever there is not a cell, we assume that there is a source of oxygen. So this is here, for instance, source of oxygen. Well, I say source, but if there were no oxygen in the blood vessel anymore, which will never happen, but for the sake of mathematics, it could have been actually a sink as well. Imagine that there is no more oxygen coming in, all is equal to 0. As you can see now, the terms becomes negative, and now the oxygen is leaking by this point. But this will never happen, but it's just for you to understand the kinetics. So in the last term, minus alpha CIG is the uptake of the oxygen by the cells. So if you do have a cell at point IG, CIG will be equal to 1, and basically the minus alpha will just be the uptake, so the consumption of the oxygen by the cells, because the cells do need oxygen to live. And now what you can do is just run this. So I'm skipping completely the numerics, also because I didn't do that, but basically this was programmed in C++, using finite differences. Basically what you can do is play a bit with the shape of your tumor. So here, case number 1, my tumor is furry, so all cells are packed together, which is the most classical scenario. And here, my cells are represented in the first column, and in the second column you will have the gradient of oxygen. So whenever it's black, sorry I missed the scale, but whenever it's black, you don't have much oxygen. Whenever it's blue, you do have plenty of oxygen. So as you can see, basically, whenever there are cells, the cells are actually consuming the oxygen. And they are consuming the oxygen a lot, more than the blood vessel can actually provide. And this is why you here, you have this darker colors when the cells are. And on the opposite, when the cells are completely spread apart, which could happen in some types of cancers, now the blood vessels, if you can imagine in our domain here, the blood vessels will be also spread apart again, because whenever there is not a cell here, there is blood vessel. And here basically we are able to, the spatial configuration make the oxygen available for most of the cells. Okay, so now remember that pH was really important for temozolomide PK. So the next step was to model pH. And it's actually quite complicated, so we have decided to go with the simplification. So we have used a very simple equation, which may not be true in all cases, but which holds some sense. So here, basically, so this is the map I just showed you with the oxygen level, and here this is drawing the oxygen level for one cut like this, horizontal cut. So I is just here this coordinate, horizontal coordinate. And we are looking at the oxygen here. And basically the idea is that we don't want the pH to go below some physiological threshold, and we don't want it to go above some physiological threshold either. So when oxygen is above some threshold, which is this line actually, then we have this formula for the pH, which can go, as you can see, it can go above this pH max. And whenever the oxygen is really low, the pH will never go below this pH mean. And the delta pH will be pH max minus pH mean, and same for delta oxygen. And typically, the pH max will be the pH that you can see in healthy cells and healthy tissues, which is around 7.4. Then if you measure your pH in your body, you have 7.4 pH. And the pH mean is typically the pH that you expect for tumors, and we expect it to 6.5. Okay, so this is a very simple equation. As you can see, not really mechanistic. And this is what you get when you actually run this in the simulations. And as you can see, the pH actually follows the oxygen level, which is what's expected. Okay. So now the scale of the pH here goes from 6.6 to 7.4. And as you can see, when there is no oxygen, the pH becomes acid. So low pH values are mean acidic environment. And here you have the green physiological normal pH. Okay, so up to here I have my tumor. Okay, I have my model for my tumor. Now I want to add the model for the drug. So I'm going to use actually more or less the same model that I developed in the OD part. So first of all, I want to address, or I want to show you the equation for the extracellular temozolomide. So extracellular temozolomide, we also follow a reaction-diffusion equation. So again, just like for the oxygen, the first term is diffusion. So when you put temozolomide somewhere in the domain, the temozolomide can diffuse with a constant D. And then here, the three next terms are exactly the mathematical model that we derive in the in vitro situation with the ODs. So the first term here is just the activation of temozolomide into MTIT. So remember we have this KT, so it was a small k, sorry. No, it's a capital K. So this is activation. And this happens whenever there is a cell or not, okay? Because it's just spontaneous action. On the opposite, the two next terms are actually the uptake and efflux of temozolomide in the cells. Meaning that this will happen only if there is a cell. So basically, if your coordinate x and y belong to C, where C is the ensemble of cells, or you can say that you can also define C as the ensemble where Cij is equal to 1. Okay, so just to repeat, diffusion, activation, transport towards antiroid cells. So I would say same as in vitro except diffusion. Okay, so that's for the spatial department, or the spatial model. And now, here, the second part is for the ODs. So at each node, we actually run the OD model that was designed in the in vitro study. And here, this is exactly what we have. Temozolomide intracellular can, basically temozolomide can go through the cell membrane. Then temozolomide is transformed into MTIT, and then MTIT gives rise to the cation, which will then attack the DNA. And this is just a term we have to add because it's just a spontaneous reaction of this species, which is a super reactive species. Whenever it is present in the medium, actually it's going to react very, very quickly. and with any synthesis around. So basically this reaction is really rapid, and this one is the most important because this is where the DNA will be imaged. Okay, so let's run this. So we run this for the domain I just showed you. And the first thing that we wanted to look at is how the vasculature, how the blood vessel network will actually impact on the drug availability. So here, basically, we have a tumor, and we have computed the extracellular concentration of temozolomide for two different cases. So the first case is basically when the drug is homogeneously distributed at time t equals 0, which is basically when you have plenty of blood vessels around, and so the cells can see the drug at the beginning, time t equals 0. The second case is when the blood vessel network is damaged, and basically the drug will be delivered in the border of the tumor and will not be delivered in the center. So this is time t equals 0. This is when you put the drug. And this is to represent different blood vessel network because the drug comes from the blood vessel. And as you can see, when you give the drug uniformly, the temozolomide can actually enter the tumor. Maybe I should have said that bright green is when the drug is here, and white is when the drug is absent. And on the opposite here, what you can see is that over time, if the drug is not given inside the tumor, it will never manage to actually reach the inside of the tumor. Because you can imagine that basically the drug is actually activated very quickly. This term here, minus K6C0, you may have seen that the kinetic is quite rapid. And basically in the simulation, what we see is that the drug is activated before it has a chance to actually diffuse towards the center of the tumor. So this is the first mechanism of resistance, basically. So basically, if you don't have blood vessels in the tumor, what the modeling tells you is that you will never be able to reach the cells. So you can basically never kill the cells in this situation. And if you actually draw the different quantities of the model for 3 cells, let's say I have 3 cells, so here at the center, here and so yeah, here at the center, then intermediate, then border. And then I want to, if I draw the different concentrations, so there will be temozolomide outside the cells and inside the cells for the different cells. So actually the middle blue and the dark blue are actually superimposed. This is why you don't see them. But basically what you can see is that in the case of homogeneous capillary network, so capillary means small blood vessel. So for an homogeneous and not damaged blood vessel network, you will get DNA image here. So DNA is DNA image, it's an efficacy marker. You will get some DNA image for all cells. On the opposite here, when you have a damaged blood vessel network, you will not get any image, any adducts at the center of the tumor. And then what we have done is to run the same type of numerical simulations, but for different shape of the tumor. So this is basically what I showed. For the spheroid, you need to see this, to read this from this is the initial time until 8 hours of exposure. And basically, as you can see, when you have a smaller spheroids, you have the same wavelength. Things are going basically in the same direction. So in the center of the small spheroids, you don't get any drug, hence any DNA image. And on the opposite, if the cells are spread all over the domain, then you are able to reach them. Okay, so the next thing that we wanted to and this is just averaging. If you want to average all DNA image for all cells, for each of the three scenarios, this is what you get. So basically the only possibility or the only case in which you are able to actually have a high image is the green one, so the one where the cells are spread. Okay, so now we have a nice model with cells which are described individually. So the next question was to study a bit the inter-cell variability. So we want to make the cells a bit different, okay? And the idea is to actually put the difference between cells in the transport of temozolomide. So remember we have this intracellular body model, and the idea is to make cells a bit different by changing the parameters PT and PT2. So now you have to imagine that for each node you have different values of PT and PT2. So the cells are not the same anymore, okay? So, and the way that we have chosen PT and PT2, so PT is for the uptake, so going towards the cells, and PT2 is for the efflux, going outside of the cells. So the way we have chosen PT and PT2 is just sampling them uniformly on specific domains, which is basically having an 80% deviation from the reference parameter. And the reference parameter is the one that was estimated in the in vitro setting. Okay, so we do that, and then we run again the simulations. So now again what I'm showing is the DNA image here, but now the colors are not the same, be careful. The colors are like this. So the blue is the former case where there is no variability in between cells. Cells are all the same. The red is when you have variability between cells, but only for the efflux, for PT2. And the green is when you have variability among the cells, but only for the uptake. And basically what you can see here, and this is the zoom without the error bars. So first of all, what you can see is that the error bars are super large. Okay? So depending on the parameters PT or PT2, you will have very different image in every cell, which may not, which also depends on the position of the cell. But now, as you can see, basically you do have a bit more of image inside the tumor as compared to before. And what is important to see is that just putting heterogeneity between cells, this is the only thing that we have changed. Right? We have just put heterogeneity. We actually change the average level of image. Okay? So this is important to basically understand that if you assume that all your cells are the same in your model, you will not get the same results as if you assume that the cells are actually different, which is the reality. Again, not all cells are the same, you have stochastic effects and so on. And here in this particular setting, with this particular parameter values, what we see is that basically just adding heterogeneity in the efflux of the drug, you will get more DNA image. And this is a bit surprising, right? Because you don't expect this, because on average the PT on average you have this blue line, and if you have if you have the reference PT to PT2, you have this blue line, and if you have varied PT2, you get this red line. However, I'm not showing this, but you can go in the publication if you are interested in that. However, this is important. This result only holds for the reference parameter values that we have chosen. So those PT plus and PT2 plus. And if you change that, so basically if you change your domain, you will actually change these results. There are cases where the red curve can actually go below the blue curve. So this is this result, so that the heterogeneity in efflux increased damage is actually very dependent on the reference parameter that you take, so PT2. And this is important because basically the idea is to say that if you don't have a good idea of your kinetics parameter, you may actually conclude something that is completely wrong, because those models are actually very sensitive to parameter values. Okay, I will just finish this part by saying that this was a 2D approach, but you can actually go 3D, and this is what my colleagues have done. I will not show this, but I would like to mention this very nice tool if you are interested in spatial modeling. So this is called PhysiCell. This was originally developed by Paul McLean, who is now in Indiana University in the US. And so this is a framework that is available for everybody that will basically provide you with the code, mainly C++, where you can actually have this setting to participate of 3D simulations, and you can put your ODE model inside each cell. So basically they provide the thing that corresponds to the spatial ODEs, and as well as the cell actually drawing, which is something that we have in our model, and the cell slide. And if you are interested, just go and have a look to the website and also to the publication. Okay, so just to conclude this point, so what we have done is that we have extended the cellular PK study of temozolomide by adding the spatial dimension into D. What we have seen is that the distribution, the spatial distribution of the drug may actually highly impact its efficacy, and actually just the shape of the tumor may be a reason of the drug not being efficient. And what we have seen as well is that including inter-cell variability can actually change your results, but this is very dependent on the parameter estimates, parameter values that you actually have in your model. So it's not like a proper theorem that you could infer. That's more complicated than this. Okay, any questions about this part number 2?
What is the software that you use for the simulation?
Okay, so the question is what is the software that we have used? And the answer is that my colleagues, so I should mention them, so it's Angélique Stéphano in the University of Grenoble in France. So they have actually programmed everything from scratch. So they program in C++, and they actually had something, a mathematical model that was already developed for another project, because they've been working on this for a long time. So they have programmed everything from scratch using C++. But to be honest, if you are interested in that, go and see PhysiCell, because PhysiCell is actually quite handy and very powerful. Other questions? No? Okay, so if not, let's go to the point number 3. So everything was PK for now. Okay, remember PK is what the body does to the drug, transport, metabolism, so on. But now we would like to describe the pharmacodynamics of the drug. Why? Because remember that what we want is to actually identify drug combinations that will kill the glioblastoma. So now I'm going to go into the pharmacodynamics of the drug, which is very complicated. And I'm going to, this is warning you, the model is complicated, I'll try to guide you step by step. Okay, let's start by the beginning. So this is the beginning of the model. So here you have the PK, you should recognize that. Okay, temozolomide, MTIC, and then the methylating cation. So now this cation, as I was saying, will react with the DNA. And it will create two types of DNA image. So it's called DNA adducts. So the two types are this OSIS MG, and these ones, which are three types that we actually lump together because they react in the same way. And this one, the DNA image can be repaired. Anything in green will be repaired, okay? So this one here can be repaired. And so basically what I'm showing this is that, let me draw this. So you have the DNA with the bases like this. And let's say you have a G here, a C here. Basically what the cation will do, it will attach something here, which is a methyl group, and it's like this, methyl. And what the MGMT enzyme is doing is that it's actually taking this out, okay? So without ME in the model that is called O6G, and with ME it's called O6NG, okay? This is really important, this MGMT, please keep it in mind. Why is it important? Because plenty of patients actually overexpress this MGMT. So you can imagine that if you have plenty of MGMT, the repair will be super efficient and basically there will be no damage, hence no efficacy. So this is the equation here. Well, I will not go through the details here, but the idea is that you have this CIN, which is this cation here, CIN. So this CIN will create DNA damage. Here, this species, as you can see, it's not here because we have seen that this is in excess. So basically the amount of available O6G is very, very large as compared to the amount of cation. Hence, you can say that this is constant, and this is what we have done. This is a typical trick. When one of the species of the law of mass action is in excess, you can actually say that the law of mass action will not impact the total amount. So this is this term here, so this arrow. Then this arrow is for the KMGMT, so this green arrow, so the repair of the damage. And here, this term is the one that you don't see yet, but you are going to see it in a minute. This O6-methylguanine, so sorry, that's the full name, O6-methylguanine, O6MG, will then be transformed in something else. And this part, basically, you have the same. So you have the image here that is repaired by something, and that's it. And just to give you an idea, basically, what is funny, or funny, I don't know, but what is strange is that here you have like 85% of the DNA adducts that will go here, and only 15% will go here. And yet, for patients who do not overexpress MGMT, this is the most active part. So meaning that this one, we know that this arrow is actually very fast, but those guys, the BER, which is a type of repair, R is for repair, BER is super active. We know that it's super active, super efficient, meaning that this part of the model, in normal situation, we shouldn't actually see something from here. Okay, so now, just downstream of that, the cell cycle will impact the pharmacodynamics. So I don't know if you're familiar with the cell cycle, so I just put one slide to show, or to remind you, I guess you probably all know what it is, but just to remind you, because this is an introduction. So this is how the cell population will grow. It's one cell that becomes two, basically. And you have different phases. So you start in G1, and basically in S, this is synthesis. This is when you synthesize your DNA. From one copy of DNA, you will go to two copies. And then you have G2 and N. And why is it important here? Because some things in the model will only happen in the S phase. So the cell cycle is an area where there is plenty of modeling. Maybe you are aware of it, I mean, if you are interested, we can discuss this. There are plenty of different models about the cell cycle at different levels. But here, really, the only thing that we need is a marker to say, am I in S phase, yes or no? That's the only thing that I'm interested in. That's the reason why we decided to go with something very simple. And this is what I'm going to show you now. So careful, that's the competitive model this week is coming. That's it. So that's the whole model. So this part here is what I just explained. And then I'm going to explain a bit more the rest. Before all this, maybe just focus on this module here, which is a cell cycle. Let me just explain this first. So the idea is that we want a marker for the S phase. And we know that the cell cycle is actually happening in a 24-hour fashion. So what we have done is that we have decided to use a simple cosine, and then to say that we are in S phase at some part of the cosine. So I think that we have said that basically the S phase will occur at the peak. So this is time. This is actually CQA, and basically we say that we are in S phase whenever we are around here. So if I just put my marker, it's more or less that. Okay, this one is my S phase marker. Okay, that's it. So now I guess you can just have this in mind. This is an S phase marker. So now the idea is that basically O-cis-methyl guanine will go through several steps which I'm not going to describe. Anything in green is repair. Then here you go to double-stranded breaks, so breaks in the DNA, which are also repaired by HR. R is also again for repair. Yeah. And then it will activate ATR, which is a good, a well-known activator. And then at ATR, we actually activate check1 that will inhibit CDC25, that will then inhibit, and that's the interesting part, cell cycle. So I should have said first that the cell cycle, as you can see, the cell cycle is actually needed at two steps here. Okay, you can see the arrow here and the arrow here. So basically, what does it mean? It means that if you are not in S phase, if your marker, the S phase marker is not equal to 1 but is equal to 0, you will never pass those steps, those two steps here and here. Okay, so you basically need to be in S phase to pass those two steps. Okay, and actually this will go to 0. So basically, imagine that here this is activated, this will be inhibited, and then this will inhibit the cell cycle, and you will see something like this. Now the cell cycle is going pretty fast. Okay? You don't oscillate anymore, you just stay to 0, and your S phase marker will always go to 0. Okay, so that's the blue part is ATR check1, and the last part is P53. How many of you know P53? Nobody? Okay, so P53 is like a super important protein. So P53 is like a super important protein that is mutated in half of the cancers. And it's called the guardian of the genome, because basically it's the protein that is able to trigger apoptosis, which we have seen in the first lecture, when the cell is damaged. So basically this is really a guardian in the sense that if the genome, if the DNA is damaged, then it will say, okay, this cell is not good enough, it's actually dangerous for the body, so I'm going to trigger apoptosis. And so this is what it is here. If P53 is activated, then it will actually trigger the cell death. And as you can see here, I have another module for the cell population, just like for the apoptosis model. So we are going to connect the intracellular model, the OD model, with the population model. So here the population model will be, sorry, I should have said this. We want to, yeah. So this is just to show how we actually model here the cell cycle inhibition. So this is the S phase marker basically. This is the S phase marker. LCC, the exponent is like super high. And basically when you, it goes like this basically, that's the marker. Okay? And this is in terms of, CCNA is actually CCNA. And basically that's its function. It's a field function. And we do assume that things are occurring a bit spontaneously, even in the absence of cell cycle, because we do have this one that will be equal to, and sorry, this is the opposite direction, right? Here. Is that correct? When the CKA is equal to 0, this should be equal to 0. the cell population model. Okay, so if you look at the first equation here, this is something that you should recognize. So what we say is that in terms of cells, they cannot grow because we know that actually the drug stops the proliferation. But so they can only die. So as you can see, N is the cell population, the number of the cell population. Number of cells. So they can only die at this rate, K above F of T, which I'm going to explain in a minute. So it's always a problem to define how to actually connect the intracellular models to the cellular models, because it's something that is not completely physiological. And the way that we have done that is by using some experimental data, which is here. So here, one line is equal to one cell, okay? And the line will stop whenever the cell actually died. And the line in red are the ones for which the cell actually died. And the blue are the cells which are still alive at the end of the experiment. And x-axis is the time, and y-axis is actually the AUC of P53. So P53, remember, is this guy, okay? And basically, as you can see, if P53 accumulates in the cells are going to die. Those are this guy. And if the P53 accumulates, but in another manner or in a smaller amount, basically the cells are going to stay alive. And you can actually draw this type of line, which we model by this log logistic function with three parameters. Then you can separate between cells which actually die and cells which actually stay alive. So this equation is for this black curve. So we do have three parameters. We tried actually less parameters, but this was not going well. So we basically chose this value. And now the question is how to put that into the test rate. Okay, this rate here. And the way we have done that is using again a hill function to say that basically we want the AUC, well, the integral of P53, so this quantity, which we do have in the model, so that's nice, okay? So you have the integral of P53 over time. And we want this not to go above this threshold, but as you can see, the threshold is moving, okay? The threshold is dependent on time here. And it's dependent on time following K of T. So now the threshold which is here in the field function is exactly equal to K of T. So this is saying my death rate is non-zero whenever the integral of P53 is above this black line. Okay, does it make sense? Okay, so that's basically the model in itself. As you can see, it's actually quite large model. So basically it has 62 parameters, which is a lot. And this is where the hard part is starting, because then we want to estimate those parameters, or at least some of them. And so we went through the literature first, and then we actually performed some experiments ourselves. And we were able to first to estimate 30 parameters almost one by one, which is the way that you would like to do, right? I mean, if you go one by one, the error is reduced. And this is what we have done. I'm not going through the details. And then, for the next 32 parameters, we estimated them all at once, using a lot of data, which I'm going to show partially. And now I would like to present the systems so that we are going to study. So here we are going to focus on a cell line, which is LM229, so which is a glioma cell lines. And actually we have two different cell populations. So here it's not cancer and cancer. Here those two populations are cancer, but the difference is that the MGMT minus cells are actually sensitive to the drug, they actually die with the drug. And MGMT plus cells, they are resistant to the drug. And you may actually guess why they are resistant. It's because they overexpress MGMT. Remember this guy here? And basically this is completely stopped. I mean the model here will not go further. And basically here you have 0 deaths because this part here is not working. And as I was saying, this part here actually don't go in the normal situation because this guy, the BER, are super strong, super efficient at repairing this cycle image. Okay, so we do parameter estimation. Again, Lisquare approach, CIMPA. I was thinking this like in 2 minutes, but it took 2 years. Okay, so it's really a difficult part to like gather all data and then to adapt the mathematical model and so on. And those are the results. So I'm showing some plots that we have. So here, be careful. The x-axis is the dose, temozolomide dose. So here, for this slide, I'm just showing dose dependent fit. And as you can, so the points are the data. And as you can see, the fit is quite good. So the 2 different colors are for the different cell lines. Okay? So remember that the blue is the sensitive cell lines, and the orange-ish or red are the resistant cell lines. And for instance, for this OSIS MEG, we have some data with respect to the dose. And for the DSV, the double-stranded breaks, we also have some data, which of course increase when you increase the dose. We also have some data for P53. So this one is the P53 not activated, this one, and then this one here, the P53 activated is here. And we also have, so as you can see again, a nice fit. I should have said that we do have, so for here we do have 2 exposure duration, because here we have 4 curves. Here we have exposure duration at 24 and 72 hours. And finally, maybe let's go directly to this part. What we can also use the cell model to fit this type of data. So here we have the number of cells which are actually dying. And here we have the number of cells which are surviving. So you could say it's a bit the same, but actually in terms of biology it's not. So those are 2 experiments. And this is why we have 2 different sets of data. And so what you can see here basically is that when you increase the dose, so this is a logarithmic scale, when you increase the dose, you have this typical shape S shape for this curve. Probably some of you have already seen this type of curves. And basically for this you have two values that is important. The first one is the IC50, we call it IC50, which I'm going to talk about a bit later. Okay, so this type are usually modeled also by log logistic. They are log logistic curves. And here you have 100% should be here. And this, whenever you have 50% of survival of cells, this value, so this is temozolomide, or any branch, this value is called IC50. And the idea is that you want to track the quantity of cells, the quantity of drug that you need to kill 50% of the cells. This is a typical parameter that the pharmacologist use. So you have this IC50, remember the definition, and the next one, I guess you know it, is AUC, area under the curve. So I'm going to talk about those 2 parameters later on, so probably better to. And as you can see here, so the blue is for the sensitive cells, red is for the resistant. And basically, as you can see, the IC50, here it's actually quite low. It's remember the quite log scale. Here it's below 10. And on the opposite here, so you need basically less than 10 micromolar to kill your cells, 50% of your cells. And here, you're not around here, so which is around 300 or 400 micromolar. So as you can see, this is not really resistant because this type of concentration is completely unrealistic. Maybe just to give you an idea, the patients they will see this something around here. So in the blood of the patient, you have 20 micromolar of temozolomide, more or less. Okay. I guess you are now convinced that the model is reproducing the data at least in a dose-dependent manner. We also have plenty of other plots in a time-dependent manner. I will go through them very quickly. So here now the x-axis is the time, okay, not the dose, time, and the y-axis are the different model values. And so this is temozolomide and one type of adult. MGMT, for which we do have data. So as you can see, MGMT is equal to 0 for the sensitive, and to some things that we actually did not manage to fit for resistant cells, and this is really high in terms of protein concentration. Okay, and then maybe what is interesting is that you do have an effect here of the cell cycle. Remember that the cell cycle here is a cosine function. And remember that the creation of DSV is dependent on the cell cycle. So here what you are seeing is that basically for some particular time when the cells are in S phase, the DSVs are actually created. In other times, like around here for instance, the cells are not in S phase and the DSV are not created anymore. So this is just the result of the cell cycle of the cosine function. And if you actually, in the second slide, I do have a cell cycle, so that's the cell cycle. And as you can see, this is the RS of the cell cycle over time due to the drug in blue. So the blue is the sensitive cells. So I should have said that the two colors are two different doses of temozolomide. And that's it. I don't want to go into the details. As you can see, everything fits quite good. Okay, so the next question is, okay, we have estimated our parameters. How reliable is that? Okay, so now that we have our parameters, so the idea is to have a look at the parameter distribution. And one way to do that in this setting is actually to use not only the optimal parameter set, but also the best ones. Okay, so when you run CIMPA will actually explore a lot of different parameter sets. And we compute your cost function, which is again the distance between your data and your model, for different parameter sets. And instead of just focusing on the optimal one, what you want to do is actually to take the 1% best parameter set. So of course this 1% is a bit, it's something that we have decided and it's to be discussed. And basically once you have this total those good parameter sets, what you can do is then draw the distribution of each parameter, and this is what we have done. So here you have all parameters of the model, so the color codes are for the different parts of the mathematical model, and here, so we have drawn them in their normalized way, because for CIMPA you need to normalize them. And basically, so we are searching the parameters between 0 and 1, okay? So the whole search domain is indeed 0 and 1, and as you can see, for most parameters, so especially for this one, for instance, you have really sharp distributions, meaning that the estimation is actually really precise, really accurate. It's true that for others, for instance, if you take that one, or that one, the distributions are really flat, which means that the estimation here is not really good. And we can actually relate that to the availability of data for some parts of the model. But overall, we were rather happy with this. Maybe I should also mention that from this you can compute the coefficient of variation by just dividing the standard deviation by the median value. And basically the CVs, they range from 3 to 36%, which is okay for parameter estimates. So we are quite happy with this estimation. Although that's not a proper identifiability study, those results are clearly in favor of practical identifiability. The next thing we wanted to look at is the correlation between parameters, because of course we don't want to have plenty of parameters which are correlated, because then it means that your model is not well constructed. And for this we have used a statistical approach, which is called variant-popular, which is a way to actually derive a multivariable distribution for your parameters that will include not only the distribution of one parameter, but also the correlation in between your parameters. I'm not going into the details of this, to be honest, I'm not a specialist of that, and this is if you are interested, so this is the main publication. And the idea is that so you can program this in R using this package. And you can then display some contour plot like this, where you have two parameters. So here that's Z1 and Z2. And you can draw here basically the dependence between two parameters. And whenever you have round circles like this, you can say that the parameters are independent, because basically if you take one value of Z1, you can have plenty values of Z2. On the opposite, if you have this type of really sharp distribution, I can draw this. Okay, so just to say that when you have something that is dependent, for any value of Z1 you have plenty of the values of Z2. On the opposite, when you have something that is dependent, so for instance this is Gaussian, for one Z1 you may not have you have less values, that's not really well, but you have less values of Z2. Okay, so we do that for all parameters, or almost all parameters, actually not all of them. And this is what you get. So, can you read that? Not sure. Okay, I have a zoom after, don't worry. So, you don't see it, but on the diagonal, those are the parameters. So the way to see it, to read this is, for instance, if I take, what's the pointer? If I take this here, this is the contour plot for this parameter here and that parameter here. Okay. Anytime you have, so here you have the contour plot and here you have the correlation coefficient, so it's just a mirror. Anytime you have red or pink, it means that you have a correlation. So if you just look at the colors, forget about the rest, the first thing that you can see is that, okay, we don't have much correlations. So we are more or less, we are happy because if we only have some particular parameters which are correlating. The most important correlations are around here and those are actually the same parameters. So this is a zoom. Now you can see, right? Okay. So let's have a look at three parameters. So the small k ATR, capital K ATR, and the K P46. So they are completely here. So K ATR is here, and then here and here. Okay. So now if you look at their correlation, so small K ATR is actually highly correlated with the capital K ATR and with K P46. Those two parameters are actually highly correlated between each other. Okay, so this part of the model, I haven't detailed that, but maybe just going back to the model. Just going back to the scheme. Okay, so we are around here, okay? In the ATR, we are around here. And actually, we don't have data for this particular species, the ATR. Those data were not available. And this is clearly a consequence of not having the data, but we do have this type of correlation, where basically it's a bit difficult to understand what is going on there. Okay, so that's it for the investigation of the parameters. So just to back up a bit, so the idea is that we have designed the model, we have estimated the parameters using the data. Now we have evaluated the parameter estimation quality, is it accurate or not? And now what do you think we can do next? So now we have a mathematical model which is calculated. So the idea is to go for optimization of the drug combinations, okay? Okay, let's do that. That's the part that we are going through. Sorry, I forgot. So there is another thing that we actually did. It was to evaluate the importance of the parameters. Sometimes it's also the sensitivity of the parameters for the model to see basically if some parts of the models were completely useless and could have been removed. And so the way we have done this is using machine learning. So first of all, you decide which parameter you want to study. So for us, it was almost all of them. Second, you decide the output of the model that you want to study the sensitivity on. So in our case, we are going to use the AUC of the cell survival curves. So we have done two different studies for resistant and sensitive cells. We have this type of curves, which are also which are the same as the ones that draw. And we are computed for every parameter set. We are computing the AUC here, the area under the curve of temozolomide for this particular parameter set. So the algorithm goes as follows. You start by generating a lot of parameter sets using the very popular multivariable distribution that we just had for the correlation study. Then for each parameter set, you run your mathematical model and you compute this AUC of temozolomide. Then we are going to use machine learning regressors. So in that case, it's gradient boosting. I will not go through that, but there are plenty of resources online that can help you understand this. In two words, so gradient boosting is based on decision trees, and it's close to random forest, maybe if you are familiar with that. So the idea is to pack different decision trees in order to have a proper prediction. So what do we do with this regressor? We predict TMZ AUC from parameter values. So the features are parameters. The output, what is predicted, is the AUC, which is of course a measure of the efficacy, right? And so this is programmed in Python using the famous cyclic learner library. And then once you have your regressor, you can run what we call feature importance, and in particular a very good algorithm it uses Shapley values that will assess the importance of the features in your decision trees, and here the features are the parameters. So that's it for the algorithm. I will not go through more details. And those are the results. So as I was saying, we have run two parameter importance study, one for sensitive cells and one for resistant cells. And as you can see, basically, the most important parameters or the most sensitive parameters that actually define the efficacy are related to the last cell cycle model. So anything in black would be the cell population. Black is cell population. And then the purple would be p53, and the blue would be ATR, and then you do have some cell cycle here, and a bit of transport here. So as you can see, you have plenty of different colors here, or like this especially here. And they are not exactly the same for the resistant cells. Here, as you can see, you have some parameters which are important for the resistant cells, which are not important for the sensitive. But basically what we see is that the important parameters are distributed throughout the model. Okay? We cannot say this part of the model is not useful because there is no important parameter in there. So this is a bit reassuring in the sense that we decided to keep the model the way it is because all parts are actually useful for predicting the efficacy of the drug. Okay, now let's go to the optimization. So the idea is that you have the calibrating model, and now remember that the regressor is really hard to treat. So the idea is to use this mathematical model to design new drug combination that will help temozolomide to kill the resistant cells. So the goal is to find drug combination that will minimize the AUC, because of course what you want to do is to have an IC50 here that will go towards the left. So what you want is to go from a curve like this to a curve like that. Okay? So you want to minimize the AUC. That's a bit counterintuitive, so minimize the AUC. So here we haven't done a proper optimization, we have just done research. Basically what we have done is that we have selected all the relevant parameters. So for instance, I can select the rate of repair of MGMT, and this parameter, I will change it proportionally to the dose of the inhibitor. So for me, I just said, okay, I change it from 0 to 100% of the value. So that's it. So basically, you have for each parameter, you try different values from 0 to 100% inhibition. And then what we are going to do is to apply this from the beginning of the simulations, from t equals 0. So it means that basically you give the inhibitor before giving the temozolomide. Okay? So the change of the parameters again are from time t equals 0 and will remain over the few days of the simulations. In that case, 5 days. So the first thing that we wanted to explore was can we increase the efficacy of temozolomide by just combining the drug with one inhibitor, okay? Because we want to use as low as possible, and we want to keep the number of drugs as low as possible. And so this is what we get. So this is temozolomide plus one inhibitor. So here each line is one inhibitor. I'm not sure you can get that, but so the first ones, for instance, are temozolomide uptake, temozolomide efflux. Here you have base excision repair, so BER repair, HR repair, ATR degradation, PATR degradation, and so on. And so the first panel here is for the sensitive, second panel is for the resistant. And here the colors are, whenever it's close to yellow, the AUC is super small, meaning that the cells are sensitive. And when you go to blue, or dark blue, the cells are resistant because the AUC is very large, as you can see on the drawing. And here on the x-axis, you have the percentage of inhibition. So basically 0% means there is no inhibitor, it's just temozolomide alone, and 100% means that you have the full inhibition, so your parameter will go to 0. Okay? And what you can see is that for the MGMT minus, actually we don't care because they are really sensitive, but for MGMT plus, the resistant cells, we do have some lines for which you start with blue, so you start with resistant, this is what we know. And then you become yellow when you increase the dose of inhibitions. Okay? And so, how many inhibition do you think would actually make the trick? How many drug combinations do you see which are actually good enough for MGMT plus? It's written on the sides, I mean, read. So basically all those, here, those 5 lines, as you can see, when you increase the inhibitor dose, okay, going from left to right here, you actually go from a large IUC to a smaller IUC of temozolomide, okay? So it means that basically you are going from a resistant cell or resistant state to a sensitive state, okay? So meaning that if you combine, for instance, if I take this, okay, this is base excision repair. If I combine temozolomide with an inhibitor of base excision repair, I will sensitize my cells. In particular, if I go up to, let's say, around 40% here, if I inhibit my repair by 40%, I will make my cell die when I expose them to temozolomide, just after exposing the cells to the inhibitor of base excision repair. Okay? And if I explain this using the model, so base excision repair is here, and basically what we are doing is that we are removing this repair, and we are basically opening this path. And now the image will flow here, and then becomes this type of image. And this is what is actually happening. Because remember that in the resistant cells, this guy is overexpressed, so this path is completely closed. So this is one thing. And the other thing is that remember that actually this path is only 50% of the image, and this one is 80% of the image. So here, because you do have 80%, just removing a bit of this repair or reducing it, inhibiting it, will actually make the trick and allow for some flow here to actually go towards this aspect. Okay, so that's good, but the idea was, can we maybe add another drug and lower the percentage of inhibition that was needed? Because here 40%, we don't really know whether it's actually realistic to inhibit a target at 40% of this level. So we go with temozolomide plus 2 drugs here. So here, to draw the pattern here, we have set the inhibition level to 30% to go below 40%, that's the idea. So here, let me drive you on that. So the lower triangle is the sensitive cells, and the upper triangles are the resistant cells. So here, basically, you have the parameters which are inhibited, and you have actually the same, so I should have put them down, sorry. You have actually the same here in color. So, as you can see, basically, you use any type of combination, you will kill the sensitive cells anyway. So you don't really care about this lower triangle, because you put any type of inhibition, temozolomide plus 2 inhibition, and then you kill all the cells. On the opposite here, we are interested in the yellow squares, which is where the AUC is small, meaning that the temozolomide efficacy is large. And as you can see, we have some interesting lines around here, and here, and here. Okay? And those lines are the ones which are actually bold. So they are more or less the same as the ones that we actually discovered in the slide before. So this is the base extreme repair, BER. This is HR, homologous recombination. Again, ATR and P-ATR degradation, and P53 degradation. And basically, the most interesting ones were the ones, so the yellow ones, which basically kills like a very large number of resistant cells. So what we did is that I'm going to the next slide. Yeah. What we did is okay. Now we are going to focus on three different targets. So we have chosen that one, and that one. So BER, HR, and P53. Okay, we have three targets, and we go 2 by 2. But now this time, what we do is that we vary the percentage of inhibition. So this line is go from 0 to 50% of inhibition of P53 degradation, and this one goes from 0 to 50% of inhibition of this repair mechanism, HR. And on the z-axis, what you can see is here, this is temozolomide IC50. Sorry, this is IC50 actually, this is not AUC. And so what you can see is that basically you have very large AUCs here when there is no inhibition because the cells are resistant. All this is in MGFT+, so all this would be in resistant cells. So when you don't inhibit anything, you are here. And when you start inhibiting things, you have a region here in yellow, which is a region that you actually want. And depending on so you have 3 different combinations. And as you can see, maybe driving you directly to the conclusion, is that here it's a bit better because here this part is actually, the yellow part is actually larger. The surface here is actually a bit larger as compared to these ones and these ones. The curve goes faster towards 0. So we have decided to follow up with this one, which is a combined inhibition of BER and HR, 2 types of repair. So just to sum up, the idea is to add those 2 inhibitors before exposing the cells to temozolomide. So this is the better conclusion. So we have now a 3-drug basically combination because you have inhibitor of BER, inhibitor of HR, and temozolomide. And logically, what do we want to do next? The modeling is done now, so we do have something to give to the biologists. So we go to our colleagues and say, can you validate that? Okay? Because now the model has done its job, and we have done the job. So the idea is that we have a drug combination that is optimal, or is predicted optimal by the model, and we go to the conclusion, and we go to the experiment. Sorry. And then, so those are the results. So let's focus maybe on this particular panel. So here, so this is in resistant cells. We are looking at the same type of curve as before. So temozolomide concentration is on the x-axis, logarithmic scale again, and here you have the survival of the cells. So the four colors are for different types of drug administration. And basically what you have is temozolomide alone, the darker one. As you can see, the IC50 is around here again. Then you have temozolomide plus LE1, which is an HR inhibitor, so one of the inhibitor we want. Here, unfortunately, due to the dose we have used, the inhibition is not very good. And I'm sorry, there is a mistake. I will go through this again later. Okay, so RI1 is the HR inhibitor, and then niraparib is the BER inhibitor. This is actually a drug that is currently used in the patient. So this is the orange curve, and you can see that already this has an effect on the survival of the curve. It kills more cells than just temozolomide alone. And when you combine both, this is the green curve. This is the best curve, right? Because you do have an IC50 that is shifted toward the left, and you also have an AUC that is really reduced. And basically, so this is what we want, right? So meaning that we have find a drug combination that is good, and this is actually killing the cells. And we also prove here that this drug combination is actually doing better than anything else that we have tested here. So this is of course not a proof for optimality, but this is a proof for superiority, at least for those two other drug combinations. And maybe just to give you, okay, so I haven't been through the details, but basically we can estimate the inhibition. I'm sorry, here there is a mistake, so this is 50%. So with the model, basically we estimated that we had an inhibition of BER. Sorry, I think this one was 30% and this one was 50%. There is a mistake here. I will correct that. So the idea is that with the model we are even able to estimate the percentage of inhibition of the targets, which is something that is very difficult to actually measure in the experiment. And just to give you an explanation of why it is working. So remember that what we have said is that when you inhibit base inhibitor, you open this path here. Okay? And actually what we do is we also inhibit here this repair, meaning that we further open that path. What we have seen is that path is actually open a little bit, because if you don't inhibit this, just inhibiting that does show some efficacy. But now the idea is basically to open this door here and open that door here, so that the flow can go up to the cell death register. And this is what we have found using the mathematical model. So basically, in resistant cells, this path is completely blocked upstream, because this is blocked here. So you cannot go through that path. So the idea is to use the left path here and open it with two different types of inhibitors. Okay? Okay. And the last thing I would like to show in the 10 minutes that are left is, okay, we do have a drug combination, but this is just for one cell line, okay? This is in vitro. And let's see, how can it be useful for the clinics, for the patients? Can I, for instance, derive a signature to say this 3-drug combination will work in this particular patient, okay? So it's a type of response signature, except that here is going to be like we call it functional response, in the sense that we are going to say, okay, for this drug combination to work, we need this and this and this and this to be true in the tumor of the patient. And for that, we have used a virtual population. So this is going to be the last topic of today. So the idea is to design a virtual patient population. So that's the first step. I'm going to explain how. Then for each patient or each tumor to compute some efficacy of temozolomide and drug combination. and then to try to identify the signature of response. So let's go a bit more in details. So the first part here, what I call a virtual patient, in terms of mathematics, is clearly just a parameter set. So here we are going to use the Vine copula, because the Vine copula was really just for the cell line that we have used, so we can say it's more or less for one patient. On the opposite here, we want to have a patient population with a larger heterogeneity. So what we have done is that we have used distributions, log normal distributions, with larger standard deviations, with larger dispersion, so that the parameter values can have more values than just the ones that we estimated for this particular cell line that we used for our study. So that's number one. So here we have log normal distribution. You sample from them, okay? And we sampled about 100,000, okay? 100,000. So because anyway, you can do whatever you want. This is virtual. So we have about 100,000 patients. So this is like quite a lot. I mean, in real world, if you have 500 patients, you're already happy. So we have about 100,000 virtual patients, which is more or less equal actually to parameter sets. So those are tumors, because it's true that when I say patient, actually what I mean is tumors, okay? Because we are actually modeling the tumor here. Now, I take my parameter set, I put it in a mathematical model, and this is number 2. I can actually get my AUC for both temozolomide and the 3-drug combination, so with the inhibition of ER and HR. And the question I ask is, would this patient benefit from the adding of the inhibitors? Because basically all patients will receive temozolomide, and there is no doubt for that. So the question is, if I want to help the clinician, I want him to make a decision, and the decision is, do I add those 2 drugs, BR and HR inhibitor, on top of temozolomide? So to do that, we have used as an output the difference of AUC between the curve that is just with temozolomide alone and the curve which is with the 3-drug, okay? And basically, if the AUC difference is 0, it means that there is no benefit associated with the 2 drugs, and we don't want to administer those drugs, because you could even imagine that the patient can have toxicities with this drug, so we don't want to add them. On the opposite, if the AUC difference is large, it means that you have managed to sensitize yourself to temozolomide, and you do want to add the drugs for the patient. So this is one parameter set, 2 model outputs in terms of AUC difference. And now, just like we did for the parameter importance, we are going to use gradient boosting. So we use a machine learning regressor to predict the IUC differences. Okay, now the prediction is the IUC differences, and the features from which you predict are the parameters. And again, we use Shapley values to assess the importance of those parameters. And basically, those parameters which are important are going to be our signature. And then I will cut the story very short. And this is the result of the importance. So here you have the Shapley values for the different parameters, so the ones the most important. Here I should have said that we have done the analysis for all patients, whether they are MGMT plus or minus, whether they overexpress MGMT yes or no. And basically what you can see is that the first important thing to benefit from this 3-drug combination is actually to overexpress MGMT. In other terms, basically, if you do not overexpress MGMT, actually all the damage is going through the right path, the one with cis-methylguanine, and you don't really benefit from this 3-drug combination. On the opposite, if you do overexpress MGMT, you do have an interest in adding drugs. And then you have actually 3 other things which are important, which is basically, in a sentence, you can summarize this as, okay, once I have activated, or once I have opened my 2 doors, I need the following model to flow. So I need, to go in order, I need ATR to be here and present the marking, I need P53 to be present the marking, and I need the cell death to be working. Okay, so those are the three colors here. So ATR, apoptosis in black, and p53 in purple. So basically, it means that once I have opened those two doors, I need the rest to be flowing. So I need my model to be true in a sense, so that the patient benefits from it. And so this is machine learning, and as we were saying, a drawback of machine learning is that maybe you don't have a mechanism-based approach, and it's difficult to actually understand your results. But here, hey, cool, we do have a mechanistic model on top of this machine learning. So here we went back to the mechanistic model just to have an idea about this dependence of MGMT. So what we have done is that we have created virtual patients which have all the same parameters except one, which is MGMT level. So this is on the x-axis, here you have the MGMT level. So then the y-axis on the left refers to the two lines here, which are the AUCs of temozolomide alone and temozolomide with the two drugs. And the right y-axis refers to this area here, which is actually the difference. And as you can see is that, well, as expected, the tumor resistance to temozolomide increases with MGMT level, in the sense that the AUCs here are increasing with MGMT. The more MGMT you have, the more resistant you are, which is expected. But maybe something that could be nice to see is that actually this area also increases in the sense that the difference between those curves increase. You know, as you can see, those curves are not parallel. They are going like this. They are not parallel. And basically, what does it mean? It means that those patients here with very, very high amount of MGMT, they do have actually a better benefit, a higher benefit, to have the optimal drug combination. Okay, with this I will conclude. So what have we done here in the part 3 of this talk? So we have designed a quantitative model, so QSP model of temozolomide PK and PD pharmacodynamics, and this model integrated a large, well, let's say multiple data sets which were both dose-dependent and time-dependent. So using this model with a typical QSP approach, we have designed a drug combination that is efficient and that was validated, that corresponds to temozolomide plus an inhibitor of ER plus an inhibitor of HR. And using machine learning, we have actually derived a functional signature for this optimal drug combination that further needs to be explored in different cell lines and different patients to see whether or not we can derive a proper signature to predict the efficacy of this new drug combination before even administering it to the patient. That's it for today. Thank you very much. Any questions?